All right, so we are going to get into the nuts and bolts of replication and how that actually occurs with all the different enzymes are involved and things like that. But please remember that one critical thing about the structure of DNA and how it works in replication is its directionality. We've got these number, the carbons are numbered in the sugar to help us organize and understand the molecule, and you need to remember that numbering scheme, and that the 3' carbon, which would be right here in the ring, and the 5' carbon, which is up here apart from the ring, are really important. And the 5' carbon is what the phosphate group is stuck to. The 3' carbon is going to stick to the phosphate group of the next nucleotide in the long chain of nucleotides that makes up DNA. Okay, such that what we get is, if we're looking at this one strand on this side here, we've got the 5' end of the molecule and the 3' end exposed down here, and on the other side we've got the other strand, but remember it's upside down in relationship to the other strand, so that the 3' end is exposed here and the 5' end is exposed here. So they are anti-parallel and of course complementary, A to T, C to G, going down the middle. Okay, there are a bunch of enzymes involved in just copying this DNA. Remember, we're copying it to give to offspring cells, give exact genetic information to offspring cells. There's a lot of enzymes involved in that, and we're going to look at how the enzymes and the molecule interact with each other. Now, the first step in replication is that we have to get that double helix that is twisted up. We have to unwind it. And the molecule that is responsible for that is an enzyme called helicase. Helicase unwinds the helix. So that's one way to remember the name. It is represented by these yellow wedges here that have unwound the molecule and are opening it up. Okay, the molecule really does want to twist back into a spiral shape, so there are other little proteins called single-stranded binding proteins that are represented by these little blue circles here that will kind of hold the molecule open. Now, what we get when we wedge this open with helicase is we have this whole structure where the DNA strand is open a little bit, and that's called an origin of replication, just a place where we begin replicating. Okay, and each end of that, where a helicase molecule is actively wedging it apart, each end is called a replication fork. And there's one over here as well. And it's at the fork where the actual business of copying, of replicating, is going on. Okay? And yeah, I'd love to be helicase and unzip your genes. It's an old joke. All right, replication. The second step is simply, once we've wedged it open, we can start adding complementary nucleotides to build the daughter DNA strand. And DNA polymerase 3, that's 3, DNA polymerase 3 is the enzyme that does that. Okay, so DNA polymerase 3 would come in and add a complementary G to a C, an A to a T, a C to a G, and so on. And that's great. Looks pretty straightforward. But it's not quite that straightforward, because DNA polymerase has a problem. DNA polymerase 3's problem is that it can only add to an existing nucleotide, to its 3' end of an existing nucleotide. So if this is an existing nucleotide, DNA polymerase 3 has to have that there to come in and add another nucleotide onto this hook. Okay? This is acting as a hook for me to add another nucleotide to. If I don't have this present, I got nothing to hook onto. Okay? If you look at it in this diagram, it's a little bit more clear. if we open up the DNA molecule, as I just did, so now there's this great big empty space in the middle, I need a hook here. I need some kind of nucleotide to exist here to hang my next nucleotide off of. I've got to have a 3' end here. And I don't have one. I don't have one. So I can't add anything because there's no hooks in the middle. It's empty in the middle. How the heck can you add to the existing 3' end of a nucleotide if there's no nucleotides in here? There's no nucleotides in here to add. There's non-existent. So that is a problem. So what do we have to do? We have to create a hook. And what we're seeing right here is we have created a hook. We've put a nucleotide here. So now there is a 3' end of that nucleotide, and I can hang on another nucleotide onto that, onto the one after it, and so on. So there's my template strand, there's my hook, and now I can add to that. I have to have a hook present, and then once my hook is present, I have no more problems. But the obvious question is, where the heck did that nucleotide hook come from? Who stuck it on there? Okay? The answer to that is that the hook is actually called a primer. It's a little piece of RNA. It is a little small bit of RNA. It is not DNA. That hook is made out of RNA, so it's not really proper DNA material. And it is put there by an enzyme called primase. So primase lays down the primer, okay? And primase is the only enzyme that can add any kind of nucleotide, DNA, RNA, whatever, just directly to an exposed base like this one without having a 3' hook already present to hang off of. So it doesn't need a 3' hook to be existence over here. It can just kind of plop something in right there, a G next to that C, and now there is a 3' hook from which we can hang bunches of other nucleotides going on down the chain. Okay? So that's really important. The primer, the hook, allows us to complete replication. But why do we need that? Why do we need that hook? And yeah, shut up. I know nobody cares. Leave me alone. It has to do with the way the bonding of one nucleotide to the next in a chain is energized. Now, don't get bogged down in this, but what does energy for anything in biology usually come from? You ought to recognize that as that is an ATP. There's your A. We don't really care what this part of the molecule looks like, but when we see that, we think right away ATP, okay? Now, if we want to get energy from that molecule, we know that we just break off that last phosphate group and there's energy and we have ADP left over. But that's not the only way to get energy from ATP. We can also get energy by breaking off 2 phosphates from here, and that leaves us with something called AMP. Well, if you look at that, that's a base, adenine, sugar, well, that's just an adenine nucleotide. The thing about these nucleotides is basically they exist like this when they're by themselves. And by cleaving off 2 phosphates, they provide their own energy to bind with other nucleotides to make a DNA molecule. They bring their own energy to do that. The cell does not have to provide energy for that to happen. The molecule comes with its own energy. We just cleave off the phosphate groups and then leave a nucleotide on the DNA molecule. Now, are there other energy nucleotides? Sure. Here, let me clear off this slide real quickly. Whoops. Okay. GTP exists in the cytoplasm as like this, but if we cleave that off, then we're going to get GMP, which is just guanine. Thymine, same deal. Cytosine, same deal. So, DNA nucleotides come with their own energy for making a bond. all we have to do is cleave this off, and then the bonding can take place right here at this site where that energy is cleaved. But it does have to take place right here, okay? And that's what's going to limit us in how we can build a DNA molecule, okay? DNA polymerase brings molecules, nucleotides, to the primer here. And if you'll note, the primer, there's the 3' end of this primer, and we're going to add to that with DNA polymerase. We're going to bring in a nucleotide, but remember, we're going to cleave off those phosphates so that the energy is provided to create this bond here between this nucleotide we just brought in and this primer nucleotide that already existed. And we're going to do the same thing when we bring in another nucleotide. We cleave the bond, and that energy will connect these two nucleotides right there, creating a covalent bond, and so on, and so on, and so on. But that has to happen at that 3' end. That phosphate has to cleave next to the 3' end so we can leave that covalent bond there. All right? So, I'm going to come back to that. If we talk about the other side of the replication fork, however, the other side, it's going to be a little different. Let's look at this. All right, I'm going to repeat for you. Notice the labeling I've put in. This was the original DNA strand that was together. We've just opened it up here. And this is the 3' right there, the 5' end here, 5' end here because they're anti-parallel, 3' end down here, there's the 3' right there. Okay, so I'm going to put a primer on here because, again, primase has to come in and put that first nucleotide on, and then I can just bring in my nucleotides and attach them, and it's very smooth, very smooth. We just go straight up as far as we can go, and it's a very continuous path. As you can see here, this replication goes very smoothly. But on the other side, on the other half of the DNA strand, okay, if I bring a primer in and stick it here, please note, what is exposed for me here is the 5' end. I can't attach to that if I am DNA polymerase 3. Okay, why not? Because this is where the energy for the bonding is going to be provided, but I need the bond to occur between those two molecules. And that's, the energy's not present at that point, it's present up here. So when I cleave off those, I don't get a bond right here. So that is not going to work. So what will work then, okay? What is going to happen, and let me clear off this writing just a moment, okay, is that I need to place my primer in a different spot. So here is my 3', and what I can do is I can start growing my strand in this direction, okay? I add it, and see, I cleave my phosphates, and there's the energy to create that bond, and so on right there. Well, what about these guys? Well, what about these guys down here? Again, I'm going to have to put in another primer right here, and then there's my 3' right there, and so I can add to that. Now, if you'll notice, I have a problem because there's no bond right here. Right there, there's no bond, so I have to have another enzyme to knit that final bond together, which it will, and it's called ligase, and it links up those two right there and creates a covalent bond. Now, if you'll notice, one half of my double helix has a very smooth, no problems creating it. We put one primer in, and we just attach onto the 3', 3', 3' all the way down. But this side's going to struggle because we've got to start at the 5'. We got to put a primer in and go from 3' down to. We've got to add on to this 3' end, 3' end, 3' end this way, but that leaves this exposed and so on. So let me explain why this is a problem. Now I can go back to this other slide that I want to show you. Hold on. Sorry, it's just taking a little, there we go. All right, now this is what I was just showing you, only it's a little more diagrammatic. Here I have that where I put in a primer. Let's say this is my primer right here. This is my replication bubble. This is my replication fork right here. And I put in my primer. And then 5' at this end, 3' is right here, and I add this nucleotide to that, this nucleotide to the 3', 3', 3', and this is a 3' end down here. And as long as my helicase is unzipping me, I can keep adding to exposed 3' ends all the way. But on this side, okay, this is a 5' molecule, which means this guy has to have a 3' molecule, and thus I have to add going in this direction. So I put a bit of primer here and work out this way. I put a bit of primer here and work out this way. I put a bit of primer here and work out this way, 3' end exposed. But I have to wait now. I have to wait for this to open up further, and then I can add another primer and work out, and then I have to wait for this to open more, add another 3' end and work out this way. That's why this side gets created in little bits and pieces, because we keep having to wait for the fork to open to allow me to kind of get back in here and then work out this way. And so we have what is called a leading strand that gets made very easily, and this side is called a lagging strand because we have to kind of keep waiting for the replication fork to open up so we can keep working out in this direction. Okay? Sorry, guys. All right. And that's, again, the leading strand and the lagging strand. Okay? So only one primer is required for synthesizing the leading strand. However, the lagging strand, each new little piece that's created must have its own primer. And remember, these primers are made of RNA. We can't just leave RNA in the middle of that DNA. We do need to go in and pull out that RNA and replace it with DNA. So we've got other enzymes that we have to quick finish up our replication with. So there's a different DNA polymerase called DNA polymerase 1 that removes that RNA and replaces it with DNA. So DNA polymerase 3 is the main enzyme, but 1 comes in and takes out the RNA and replaces it with DNA. And then ligase is going to make that final stitch between two fragments that were once separated by that RNA primer. Okay, this is a nice diagram showing you, again, the leading strand being made very continuously. This is called, by the way, continuous synthesis of DNA. This side is called discontinuous synthesis for obvious reasons, because we have to wait for this to open, work out a little bit, primer, work out a little bit, work out a little bit, and we keep waiting for this to open. The little pieces of DNA that are made are called Okazaki fragments, after the guy that discovered them. DNA ligase is the enzyme that will knit these spaces together that are created between the fragments, and so on.